All right, we'll get going. Hello and good morning from Oregon. Welcome to the March 2023 installment of our GCE for All International Webinar Series. Today, we are very excited to have two world-renowned scientists, Dr. Stephan Pless and Catherine Lang. First up today is Dr. Stephan Pless, Professor of Molecular and Cellular Pharmacology at the University of Copenhagen in Denmark. Dr. Pless earned his PhD with Joseph Lynch at University of Queensland in Australia in 2008, after which he postdoced with Chris Ahern and Harley Kurata at the University of British Columbia until 2013. He started his independent career in 2014 at the University of Copenhagen in Denmark, where he became full professor in 2020 and continues to make substantial advancements in our understanding of ion channel structure and function. His lab employs a wide variety of biophysical, chemical biology, and protein engineering tools, including genetic code expansion. Particularly exciting last year was the solving of the structural architecture of the human sodium leak channel, which regulates the resting membrane potential of neurons to modulate respiration, circadian rhythms, locomotion, and pain sensitivity. He has been prolific in publishing and getting funding, having too many awards and accolades to list off here, though I will mention recent major funding that he obtained from the Lundbeck Foundation. So thank you, Dr. Pless, for joining us today, and please take it away.
Thank you very much for the kind introduction, and thank you very much for having me today. I very much look forward to sharing with you some of the work that we've been doing on sort of identification of neuropeptide and toxin binding sites on membrane embedded receptors. Can you see my screen all right? Just checking.
Yes, you can see it great.
Perfect. Okay, perfect. So as you briefly mentioned, my laboratory focuses on a class of membrane protein termed ion channels. These are membrane embedded proteins that open an aqueous ion conducting pathway in response to physical or chemical stimuli, and therefore regulate the cellular excitability. So my lab tends to focus on structure function studies of these types of ion channels, and we use very simple approaches such as site-directed mutagenesis. We use genetic code expansion approaches that I'm going to focus on today. And recently we've also dived into some sort of semi-synthetic approaches that I will briefly mention at the very end of my talk. Now, the first part of the talk that I'm going to talk to you about is very much about trapping peptide receptor interactions in acid-sensing ion channels and doing so in living cells. So all the credit goes here to two former PhD students, Nina Brown and Christian Borg, who really spearheaded all of the aspects that I'm going to talk to you about today. All the parts of the science, or the aspects of science that I'm going to talk to you about today are going to be focused on acid-sensing ion channels. So these are trimeric proton-gated cation channels. So whenever protons bind to the extracellular domain, they will open a cation selective pore. And the reason why we're interested in them is because we think they have a very important role in synaptic plasticity, as well as in field conditioning, but they're also quite important for the initiation and propagation of pain. So the other aspect that is sort of relevant in the context of these kind of channels is the fact that they do tend to have a role in stroke. So we're quite interested in these channels along with some other groups. Now, when we talk about the basic function of these channels, I just want to introduce to you sort of the very basics of how they work. So they can be activated by lowering the pH. So if you increase the proton concentration successively, here you see an increase in current. You should note that these currents, they're rapid in onset and they quickly desensitize. This is what we call fast desensitization. You can plot this as a function of pH and you end up with a curve looking somewhat like this. On the other hand, these channels also undergo a process called steady state desensitization. This is if you pre-expose these channels before the test pulse to increasing proton concentration, you'll see that the higher the proton concentration, the less of the response you get in response to a normally fully activating proton pulse, as you can see here. And the reason why I mention this is because it's quite striking, because at a proton concentration of around 40 nanomolar, you get full activation of the channel, whereas a proton concentration of 80 nanomolar, the channel is completely shut off. So that means that these channels are actually incredibly sensitive to protons, and this will become important later on. One of, or actually the most potent endogenous modulator of ASIC-1A is actually a neuropeptide called BigDyn. Sorry about that. It's a 32 amino acid endogenous neuropeptide, and it modulates, mostly known to modulate opioid receptors, and it's upregulated during chronic pain. But we also know that it actually targets other targets such as ion channels, such as acid-sensing ion channels. So what does it do to these channels? So if we challenge the channel with a high proton concentration, we typically see an activation inward current like this one. And if we steady state desensitize the channel prior to the activation by pH 5.6 in this case, we see a much reduced current. So this is what I've shown you before, this sort of steady state desensitization. However, if during this steady state desensitization pre-exposure, we actually have BigDyn present, the channel function is almost normal. So that means this BigDyn peptide actually prevents the channel from undergoing the steady state desensitization to some degree. And this actually has important implications because it means that BigDyn exacerbates ASIC-mediated cell death. Now, we wanted to find out, where do these peptides actually bind? And one of the defining features of these BigDyn peptides is that they are very positively charged. So there's numerous positive charges. Almost every third amino acid of the peptide is positively charged. So here's a control experiment where we show you the response that we normally get in gray. So full activation response to 5.6. However, if we pre-desensitize with 7.1, we get the green curve here, so almost no activation. In the presence of Dynorphin, this is almost rescued. But if we mutate the arginines in the N-terminal part of the peptide, for example, arginine 7 to alanine, this effect is almost gone. The same is true for arginine 9, whereas mutating, for example, lysine position 13 has much less of an effect. So we've done this for all the positive charges. And as you can see, under control conditions, you see fully desensitized channels here. This is alleviated by wild-type BigDyn, but if we mutate any of these N-terminal charges, this is much alleviated. So it means that the N-terminal charges seem to be quite important for ASIC modulation. So this was as far as we could tell the molecular determinants on the peptide. So we turn to the channel next. And the obvious place to look for the binding pocket on the channel was the so-called acidic pocket. So this is a sort of a cleft within these extracellular domains of these channels that are lined by a number of negatively charged amino acids. So we thought we look at charge-neutralizing mutations on the channel to delineate sort of the binding pocket on the channel. So what we did here, we did a control, we looked at control conditions, and then in the presence of BigDyn, we see this relief of steady state desensitization. And we did this for individual mutations around the pocket. As you can see, there was not much of an effect, actually. This was somewhat of a surprise, but when we then turned to double charge-neutralizing mutations, we saw that actually some of these completely abrogated the effect of BigDyn. This kind of hinted towards the acidic pocket being the potential binding site for these peptides, but it wasn't really quite definitive. So the question remained, where on the channel is the actual binding site? And we weren't quite sure about this. We knew we had to turn to sort of a different method to get to the bottom of this. So the obvious question here was to turn to genetic code expansion and actually try to cross-link acid-sensing ion channels to BigDyn in living cells. And so the idea being here, for example, we use something like azidophenylalanine, use the cross-linking ability of the azido group to actually cross-link this. So the idea would be to insert azidophenylalanine at different positions in the extracellular domain of ASIC-1A, then expose the cells to BigDyn, expose them to UV light, and then hopefully end up with covalently bound peptide. And I should say that this has been extensively studied by other groups in the context of GPCR, peptide interactions by Irene Cohen's group, very beautiful work, as well as transporter-drug interactions, also work from some of the neighbors here in Copenhagen. So the idea was that we really wanted to look at incorporation of cross-linkers within the acidic pocket and then also have some control sites. This was the plan. This was the work so far what I've been showing you by Christian, and it just happened so that Nina, the student who was working sort of in parallel on a similar project, she's been working on a new workflow for non-canonical amino acid incorporation into acid-sensing ion channels. And this is where the two projects converge, and I'm just going to take a slight detour here to show you what we did actually in the context of a non-canonical amino acid incorporation. So Nina, what she did was took on the brave task of actually generating over 100 stop codon or tag mutants of acid-sensing ion channels. And the idea was to use every one of them and incorporate 3 different types of cross-linking amino acids, acetophenylalanine that are introduced, as well as BZF and seleno-ABK. The idea was then to transfect these into human embryonic kidney cells. And here we would do this with the ASIC constructs, with the synthetase, with the suppressor tRNA, as well as a GFP with a stop codon, just to be able to tell which cells actually expressed all these constructs. We had to introduce a FACS step. So we did this in order to enrich the cells that actually express the GFP expressing cells. So to basically make sure that we have enough transfected cells to put on the robot, because in the end, we wanted to use a high throughput screening device, in this case, a Nanion synchro patch. And again, I should give credit here for Søren Friis from Nanion, who helped us a lot with setting this up. So the idea was to screen actually quite a few of these mutants, actually all of them, under different conditions with different amino acids in the presence and absence of the non-canonical amino acid, and then do this on at least 2 or 3 different batches. So this ended up being well over 1,000 experiments. And that meant that we really depended on this high throughput ability. I'm going to briefly walk you through the high throughput analysis here. So what we did was we did this arrow here indicates the proton application. This is just a wild type channel on the robots, and then the color coded pH challenges that we exposed the channels to. This is what the wild type channels look like. And this is a channel where the desensitization kinetics are somewhat similar, with seleno-ABK into position 236. But you can see here that there's less current and the proton sensitivity is slightly changed. If in the same position we actually insert acetophenylalanine, currents are much bigger. We get a similar pH response, but we get a much faster desensitization rate. And there were also positions where we didn't get any function. And we assumed that in those cases, that meant that there was actually no incorporation of the non-canonical amino acid. I'm going to give you an example of what the data look like. So we looked at the current amplitudes here. This is the wild type for comparison. And then, for example, in position 44, where there's usually an alanine, we incorporated either acetophenylalanine, BPA, or seleno-ABK. And you can see that they rescued the current to different extents. And we did this obviously for over 100 different positions. And you can see that some positions we didn't get any incorporation. And generally, there was a trend that acetophenylalanine worked best. We're not quite sure why that is, but that seemed quite consistent throughout the data. And we can then also look at other biophysical parameters. So we can look at the pH 50, so the half maximal proton concentration required to activate these channels. And we can see how this varies depending on the type of amino acid that we insert. So if we take a step back and look at the data from more a qualitative angle, this is what it looks like. So we have all the different positions here on this sort of dot plot diagram. And this is the data for acetophenylalanine. And the position is labeled with dark color, a specific incorporation, where we get over a nanoamp of current and less than 500 picograms of current. If we don't have the amino acid around, then we have some positions labeled here with the sort of weaker or the less prominent color that we call unspecific, where we also see significant currents in the absence of amino acid. And then we had some positions where we couldn't detect any function. And we can do this kind of analysis for all the different, or the 3 different amino acids that we actually use in this context. In the end, it turned out that acetophenylalanine was the most robust in terms of incorporation. So that's what we decided to go forward with. So this was all very good. We had now a system established where we can sort of look at sites where we can robustly incorporate these amino acids. And the next question was, can we get some sort of a positive control in our system in this acid sensing ion channel? And for this, we turned to a toxin that's called PcTx. So that's what we refer to. So it's a 40 amino acid peptide toxin from spinaverin. And it actually has very neuroprotective effects in stroke models. Now, it's a very potent and selective inhibitor of ASIC-1A. And as you can see here, normally, if we activate the channel with pH 5.6, we get a robust inward current. But here, if we challenge the channel with 1 nanomolar of PcTx1, we can actually see that the subsequent current is much reduced. So it's a very potent inhibitor of ASIC-1A. And what's more importantly, relevant in this context is that we also knew it bound to the acidic pocket. So we knew this because there was a structure of both the apostate of the channel as well as one that was bound to PcTx. We thought this was a good test system to test our cross-linking abilities. Moreover, we knew actually there was a mutation on the channel, and there's a phenylalanine position 350 or 352, depending on whether you look at mouse or human channels. If you mutate this phenylalanine to a leucine, you get a massive reduction of the efficacy of PcTx. So this is plotted out here. Usually the wild type IC50 is around a nanomolar for PcTx1, whereas when you have this F12 mutant, that's shifted by over 3 orders of magnitude. So these channels, when they carry this F12 mutant in position 350, they're much, much less sensitive to the effects of PcTx. So coming back to our story, so we had acidic pocket mutations and we had some control sites that were distant from the acidic pocket. And we wanted to see what happens when we do some cross-linking. So here we have a scenario where we just have wild type channels. We expose it to UV, 100 nanomolar of PcTx with a biotin handle on it. And then we look for biotin. For wild type, as expected, we didn't see anything. On none of the control sites did we ever see any sort of evidence of cross-linking. However, when we looked around the acidic pockets, there were quite a few sites that did cross-link, although I should point out that here the upper bands that you see in most of these cases are actually endogenous carboxylases, but these fat bands in the lower part here, they correspond to the ASIC channels bound to PcTx1. So in about half of the sites that we looked at in the acidic pocket with PcTx1 did we see cross-linking. So this was a good sign because it indicated that our system was working and we could successfully cross-link to these peptides in a living cell. One other thing that we wanted to test was what happened if we were to do the cross-linking in the presence of this F2L mutation that we knew pretty much abolished the functional effect of PcTx. So we tested this in terms of cross-linking, and we saw that although we captured a lot less, so this is basically the normal scenario of the K356 AZF mutant, we have a big fat band here for PcTx, but even in the F2L mutation, we still get a significant band that we can detect, indicating that although at this concentration there's no functional effect, there's still weak binding of the peptide to the receptor. So this meant that really the cross-linking confirms the acidic pocket as PcTx binding site. This was maybe not a big surprise, but it made us a lot more confident with what we wanted to achieve really, which was to look at whether we can actually cross-link big Dynorphin, which was the original target, obviously, to ASICS. And so these are the experiments that we've been working towards. Here again is the wild type control, in this case with 3 micromolar big Dynorphin. And it didn't matter if we used the big Dynorphin antibody or whether we used the tagged version of big Dynorphin. In both cases, this worked similarly. If we look around the acidic pocket, we get nice cross-linking in most positions. That was not the case for the control sites. And we actually looked at a bunch more positions around the acidic pockets, and most of these did cross-link if we exposed to UV. So this really confirmed that the acid-sensing ion channel acidic pocket is likely the binding site for big Dynorphin. And this was actually confirmed by a very similar work by the Gründer group, which I'm mentioning down here. So they came to very similar results as we did. What was interesting was that we did see we saw more positions that actually did cross-link to big Dynorphin than we did in the case of PcTx, and maybe if somebody's interested, we can discuss that later for the potential reasons for why that might be. So in terms of conclusions, big Dynorphin N-terminus seems to be crucial for the functional effects that we observed. The cross-linking confirms the acidic pocket as the PcTx binding pocket. And we can really also show that these cross-linkers help us to identify the acidic pocket as the big Dynorphin binding site. It also, once again, highlights the acidic pocket as a potential therapeutic target. And we're still pursuing some efforts to do cross-linking to endogenous interaction partners, although that turns out to be much more difficult than we had anticipated it to be. I briefly wanted to mention one approach that we've also been using in the lab on acid-sensing ion channels. It's also a genetic code expansion approach. It's one that's used much less, and for good reason, because it's not quite as easy to handle in most contexts. So in this context, we were interested in sort of the ionic selectivity and how ions bound to the ion conduction pathway in acid-sensing ion channels. And this was work that was spearheaded by Timothy Leiner, who's now a group leader in Bergen in Norway. And here, he was interested in sort of exchanging, for example, an amide backbone for an ester backbone. He was interested in incorporating fluorinated tryptophan derivatives or indole derivatives of tryptophan. For those at the time, there were no synthetases available, so we had to turn to a different approach to incorporate these, and this is sort of a brute force approach where you take a generic suppressor tRNA that recognizes TAG stop codon, but lacks the it lacks the terminal dinucleotide, which then comes with your non-canonical amino acid of choice, is then ligated via RNA ligase, and then you can inject this complete tRNA, including the non-canonical amino acid of choice, into your cell system. But that's the point. It requires a cell system like a xenopozoosite where you can easily inject large amounts of peptide, or in this case, tRNA. So it is very much an approach that is limited in terms of the cell type that you can use it for, but in terms of chemistry, it's amazingly versatile because you can literally append almost any amino acid or hydroxy acid to your dinucleotide and therefore do these experiments. But again, it's much easier if you work in an oocyte system and with ion channels where you have high signal to noise. Now, the last few minutes of my talk, I would like to talk about an approach that is not really technically genetic code expansion, but it is sort of an approach that we have developed or built upon work by others out of a frustration that we sometimes had, because sometimes the genetic code expansion approaches didn't work in the context that we were trying to get it to work for various different reasons. So here we turn to ion channel semisynthesis using protein transsplicing in living cells. And the work that I'm going to show you today is mostly unpublished, but it's on bioRxiv by two postdocs, Debayan and Jacopo, who work together with two master students, Sophie and Gül, on this particular project. So the background is simply that in these cases, site-directed mutagenesis did not do the trick. There were no synthetases available for the particular amino acids that we wanted to incorporate, so we decided to go for, again, yet another brute force approach, if you like, where we really incorporated an entire peptide that contained the modifications that we wanted to and inserted these via transsplicing. So the idea here in the context of an acid-sensing ion channel is we split the channel artificially in the extracellular domain, and we generate a synthetic peptide that carries the modification of choice, in this case indicated by the red dot here. And then the idea is that we use so-called split inteins. They have to be two orthogonal split inteins that would get assembled and splice out and would then generate a full-length channel that would hopefully be functional. So this was the approach that we chose for the following project, because here we were interested in a part of the extracellular domain that contained a lysine that we knew was quite important for function and was highly conserved. So it sits here in the extracellular domain, sort of in the middle part of the extracellular domain. And we had shown previously that it's likely to be important for activated, and other groups had hypothesized that it's likely to be also involved in desensitizing, potentially chloride binding of the channel. And so what we wanted to do here is we wanted to replace the native lysine with other lysine analogs, for example, ornithine, so make the lysine side chain 1 carbon shorter, use homolysine, so make it 1 longer, or use an isosteric but non-charged analog of lysine. And since we, or at the time, or at least I'm not aware of any synthetases that can incorporate these at the moment, we turn to the semi-synthetic approach to incorporate them. So this is a very simplified view of a cell, in this case, again, a xenophilus oocyte, and it's all built on some work that we've done over the past few years, where essentially we split the channel into 2 bits. So this is the N-terminal part of the channel that we express heterologously. The same thing happens to the C-terminal part that we also express heterologously. Here we had to use a little trick. We're using a sort of a faux or fake transmembrane domain to orient this C-terminal part in the correct orientation. And then we literally inject, again, these cells with a synthetic peptide. Now, I want to be very frank here. Again, the fact that we need synthetic peptide to be injected into the cell means that it cannot be used in many contexts. So it's really limiting. But in the context of these oocytes, that's very easy. And then if everything goes well, you get splicing of a full-length channel that then assembles into the membrane, and we can then conduct functional studies. In the particular context here that I'm talking about, we again wanted to insert different analogs of lysine in position 211 of ASIC-1A. Gratifyingly enough, and you can think of this as a very expensive way of making wild-type channel, if we inserted a lysine where there used to be a lysine, we got nice currents with properties that were very similar to the native channel. And we also got currents for all of the other lysine analogs that were inserted in position 211. If you look at the proton sensitivity of this, this is a recombinantly sort of assembled wild-type channel, and this is the one carrying a synthetic lysine. It's not perfectly overlaid, but it's pretty close. And actually, if we put the lysine in there, that is one shorter, so ornithine, not much happens to the proton sensitivity. It's only if we either make the carbon chain one longer, so we move the charge out by one carbon, or we remove the charge altogether, that we do see very drastic changes in the pH sensitivity. Now, the other parameter that is clearly very strongly affected is the rate of fast desensitization in these channels. So while the lysine rescue is reasonably good, it's not perfect, but it's reasonably good, ornithine and especially homolysine or the neutral analog clearly very much speed up the desensitization rate of these channels, and that's quantified here. So clearly, this sort of data indicates that the charge and its precise positioning in position 211 is involved in both activation and desensitization. So just to recap here, I've talked to you about trapping peptide receptor interactions in these acid sensing ion channels, and this was all based on genetic code expansion approaches. And then just to spend the last few minutes on ion channel semisynthesis, where we use protein transplicing in living cells. But again, just to make that very clear, that approach is much less versatile in terms of its applications. But as for us as ion channel people, this is very attractive in some of the contexts that we're interested in. And with that, I would like to thank the people that did the work. These are the former members that I've already mentioned. This is the current group, as well as our current funding that we're very grateful for supporting these sometimes challenging ideas that we're throwing at the funding agencies. And with that, I'm happy to take any questions. Thank you very much.
Okay, thank you, Stephan. Really fantastic talk and really, really wonderful science. We have a few minutes for some questions. We have one from the audience. Someone asked, how efficient is the ASIC semisynthesis approach?
Very inefficient. That's the short answer. It's not very efficient. So we haven't really quantified this in the context of the acid sensing ion channels. So based on work that we did with other ion channels, we assume it to be anywhere between 1% and 5% of sort of wild type level. So it's really taking a strong hit. Although I should mention that this really depends on the type of system that you use and where you do the splicing. So if we do the splicing like we did, can you see my mouse? Or actually, I can just use a pointer, sorry. If we do the splicing in the exocell domains, the splicing efficiency tends to be very low. So we have done splicing in intracellular domains, and there the splicing efficiency is higher. The other thing that we have to keep in mind here is that we're using 2 orthogonal split inteins. We've also, I can show you one example maybe. We have also done some recent work where we've used, this is not published again, where we use a single split intine. So this is essentially just using one split intine to insert a fluorescent C-terminus of the protein. Sorry for going fast here. And if we do this with a single split intine where we just insert a semi-synthetic C-terminus, so we just have one splicing reaction that has to happen, then the efficiency is much higher. Then we get to 20–30% of sort of wild type levels. So it really depends on the system, but again, it's not very efficient compared to what you get with many other approaches.
Great. Any other questions from the audience? Ryan.
Quick question here, Stephan. It's rare to have someone do all of these different techniques sort of on the same protein family. And because of that sort of, we'll call that protein family standard, you know, you've got a lot of valuable experience with regard to the technical challenges of each different approach. If you had to advise somebody, so I mean, we got a lot of questions in the center about like which is the best way to start. For this, if you had to say, if you were going to advise somebody to go to one of these particular approaches for putting in a non-canonical versus another, which would be your first, go-to? What would be your pecking order?
Was that car wash? Yeah, thank you. That's a very good question, actually. And it's also a bit hard to answer because it really depends on the context and the questions you want to answer. But I would say the answer has to be clearly the approach here, the classic genetic code expansion approach, because if everything goes well, so if you have a good synthetase, if you have a tRNA suppressor pair essentially for the amino acid that you're interested, then that is typically by far the most efficient way to get your amino acid into the cell. So I would say this should always be the first approach. It's only if that one fails should one venture or even think about into these other approaches, because especially this approach here, it relies on the synthesis of very large peptides and it's non-trivial. It really does work, believe me, but it's also non-trivial and it requires a lot of optimization. And the only reason why we did it was simply to overcome some of the challenges that we had with other approaches where the classic sort of approach of genetic code expansion did not yield the result that we were after. Does that answer your question?
It does. Thank you so much.
All right. Catherine, you want to ask a question?
Yeah, hi, very nice talk, Stephan. Thank you so much. I wondered a little bit if you could comment on the different photo-cross-linking amino acids that you use. One short technical question would be, why did you use the selenium-containing one? Did you try to separate spray-bait proteins for easier analysis basically of your data? And the second question, in the end, basically your acido-Phe was the winner. Could you comment a little bit? Do you think this was more Incorporation yields, or is it so much superior in cross-linking, or is it does it depend on the nature of the endogenous amino acid, maybe which cross-linker might be best?
For the second question, it's a bit of all of the above, but I would say in our case, it was mostly, and we still don't know exactly the reason. We have since tried a different synthetase for for BZF, and it has worked better. But at the time with the synthetase that we were using for AZF, I think it was just that it mostly came down to it being the best incorporation, essentially. But you're right that these other factors might also play into it. But I think the effect that I've shown you is probably an artificial, it's slightly distorted by the fact that the incorporation just wasn't as good for BZF or seleno-ABK in our particular case. But for the choice of seleno-ABK, this was basically because initially what we wanted to do, we wanted to use this for trapping protein-protein interactions and then following up with mass spectrometry. And this was basically a preparation for that, although I have to admit that so far we haven't been successful in those attempts. So we've done this, but it hasn't really yielded the results that we were after when initially designing these experiments. Also, I should say that I think we could have, or we should have been a bit smarter when designing the original setup. We should have tried to put more of the so we've been operating with 4 different plasmids, and we should have basically combined them into 1 or 2. But those were flaws that we they are on us, basically. We should have designed them differently from the beginning.
Thank you.